So first we're going to talk a little bit about dyslexia as a disorder. I'm not sure how familiar everybody is with dyslexia, the variety of theories behind dyslexia, so we're going to talk about that. Then I'm going to give you guys a quick lesson on genetics and neuroanatomy. I'm sure not all of you are neuroscientists or geneticists. And then the first statement I always get when I present this work or when I mention what I have done in the past is, you can't study that in rats. And so by the end of this hour, I'm going to hopefully prove to you that yes, we actually can. And then, of course, the question that drives all research, what can these studies tell us about the human condition? What can we learn from rodent models to then apply to humans with these disorders? So dyslexia is the most common developmental language impairment. It's present in about 5 to 10% of the population, depending on who you ask, of course. And these kids have normal nonverbal intelligence, but they have reading scores that are at least 1 standard deviation below their peers. This disorder has a lot of times been thought of as kind of a subtle disorder, but it can actually have large impacts on these people's quality of life. They can lead to lower academic success, low self-esteem, delinquency in adolescence and adulthood. There was a recent study that showed that almost half of the incarcerated inmates in the state of Texas have some form of dyslexia. And so there are many theories regarding the underlying cause of dyslexia symptoms. The one that most of you are probably the most familiar with is this visual theory. It was thought for a lot of years that dyslexia was a visual disorder, that it's diagnosed by reading, so of course there must be something wrong with vision. But if it was really a purely visual issue, people with dyslexia would be unable to copy down letters or words presented visually. So you present a word on a screen and say, just copy down what you see. And people with dyslexia have no impairments on this task. So it's not that their eyesight is bad, they can do a copying task just fine. And so this theory has kind of morphed over the years into what's called the magnocellular theory of dyslexia. And this theory states that there's an imbalance in the excitatory and the inhibitory components of the visual system. So there are 2 main cell types in the visual system, and of course this is simplifying it quite a bit: the parvocellular system and the magnocellular system. The parvocellular system is composed of small cells, and these cells focus on detail, looking at color, contrasts, lines, edges, the very fine-grained detail that you see in the world. The magnocellular system is involved in regulating the parvocellular. So as you're looking at one image and then your eyes shift to look at a second one, the magnocellular system makes sure that what you were just looking at doesn't follow you to what you're looking at next. So for example, if we were to look at the sentence, "the boy climbed up the tree," as you look at the first letter, the letter T, your parvocellular system is looking at the colors, the lines, the intersection of the 2 lines. And as you start to read the sentence, the magnocellular system is going to inhibit that system so that you get this nice, clean trace of the entire sentence, and you can read it very clearly. The idea is that in dyslexia, this magnocellular system doesn't completely inhibit the parvocellular. So as you're reading a sentence, you have this lingering trace of what you were just looking at. And you can imagine how it would be really difficult to read if you were always seeing lingering images of what you were just looking at on top of the new images. If this was the one underlying cause of dyslexia, though, it should only be present during reading and not during static text. And we know that people with dyslexia do still have deficits in naming visually presented phonemes. So although some kids with dyslexia do have visual issues, this is not the one underlying cause of dyslexia. And so the most common theory now is this auditory processing theory. And the idea being that people with dyslexia have trouble processing rapidly presented auditory stimuli. People with dyslexia require twice as much space between 2 tones in order to tell that there were 2 tones instead of just 1. And they also have problems with phonemic awareness. So for example, phoneme deletion tasks, where I would say the word break without the B sound. The answer would be rake. People with dyslexia have a hard time doing that task. Also phoneme switching tasks. So if I were to say, take the first 2 phonemes of 2 words and flip them so that doghouse becomes hogdouse, people with dyslexia also have a difficult time doing this task. And there are errors in just basic consonant identification. So people with dyslexia have almost 3 times the errors compared to non-dyslexic controls in identifying consonant sounds. So think back however many years ago it was when you were first learning how to read, and you're presented with this block of intimidating graphemes, so the symbols on the page, the graphemes. And you have to learn how to take these symbols and decode them into meaningful language. So your teacher says, this is a D, it makes the duh sound. This is an A, it makes the ah sound or the ah sound. And so you have to learn how do you look at a word on a page, break it up into the phonemes, figure out how those letters sound, put them back together into a whole word, and then figure out what that word means. So it's really an involved process in learning how to read. And it's thought that now dyslexia is a deficit in mapping those auditory representations of the sounds onto the visual grapheme. So there's a difficulty in reading silently to yourself, in essence. So what's the neural mechanism for this impairment? What's going on in the brain that's causing this mismatch? We know that neural firing patterns differ in people with dyslexia. They have a diminished mismatch negativity in the brain, which is basically the brain marking change. So for example, if you were to present the same sound over and over again, so ga, 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 initially your brain's going to respond highly. I'm sorry, is that okay in the back? I know it's kind of dark. So the first response is going to be very strong, but over time the response is going to decrease because your brain's saying, oh, this is the same thing I just heard. I don't need to care quite as much about this because it's the exact same thing as what was just played. But now if you play a different sound, so da, the response goes back up. So this is your brain saying, hey, something different just happened. In individuals with dyslexia, this marking of the change is not as strong as it is in normal controls. So the theory is that individuals with dyslexia, their brains have more difficulty marking when something has changed. They also have a delayed response latency and a lower amplitude in response to tones. So this is data from an EEG study. And I just want to draw your attention to this T4 electrode here. You can see that the dark lines are the affected individuals, the thin lines are the controls. And you'll see that the onset latency is later and the amplitude of this response is lower in the affected individuals compared to the normal controls. We know that dyslexia is genetic, highly heritable. Monozygotic twin studies have shown that there's a 68% concordance rate in dyslexia. So it is genetic and it's highly heritable. And people that have dyslexia, their brains have cortical abnormalities to them, including ectopia, which are clusters of neurons in an area of the brain where they don't belong. So, for example, this area of cortex is normal. You can see each of the dots are different neurons that are stained, so you can see them. And these neurons are pretty evenly spread out. They're not clustered together, they have some space between them. But here's an ectopia, and you can see all of the black that's clustered together. So this would be abnormal in the brain. And the genes that basically guide neurons to where they end up, they're called neural migration genes. And there are 4 neural migration genes that are commonly linked to dyslexia. KIA 0319 is the most well-studied of these genes, and this is the one I'm going to talk about today. I'm not going to talk about what it does specifically, because that's kind of beyond the scope of this talk, but if you would like to know, I'd be more than happy to talk about that as well. So we know that people with dyslexia that also have a variant in this gene have reduced left hemisphere activation, and they also have impaired phoneme awareness. This is one of the papers that I sent out with the talk announcement. This is one of the first studies to really look at the link between specific genes, neural, and behavioral components in humans. This is still very, very rare. And one of the reasons why it's very rare is that it's very difficult to say for any certainty, this gene is doing this in humans. Humans have complex genetic profiles. You're never going to have somebody come into your clinic or your lab that has a variant in one gene and no other. So it's very difficult to conclusively say this gene is doing this in humans. Lucky for us, these genes also exist in rats and mice. And so we can really get down and say, what is this one gene doing on a neural level? And so for this experiment, we did a knockdown. When you think about genetics, when you hear about genetics, you'll hear the terms knockout and knockdown, and they're very different. Knockout is basically removing the gene completely so that there's no protein whatsoever as a result of that gene being there. Knockdown is a partial suppression. So we're not affecting every single cell, but what we're doing is reducing the amount of protein that is made in the brain. And this is the model that we've chosen for this study for 2 reasons. One, because knockouts are extremely expensive to do in rats still. And 2, there was actually a recent paper that came out that showed that individuals with dyslexia that have a variant in this gene also have reduced protein in the brain. So this is a more accurate model to what you would see in the brain of someone that had a variant in this gene. So how do we create this knockdown? We take pregnant rats and we anesthetize them at a certain point during the surgery. We take the embryos from the mother and we inject short hairpin RNAi, which is basically just a bullseye for the gene that we're looking at. And then we send a small electrical current through the brain, and what that does is it opens the membranes of the cells and allows the plasmid to enter the cell. Once all that is done, we sew the mother back up, let her recover, let her give birth. Those babies are now knockdown rats. All the experiments we do are in the knockdown babies once they've already grown up. So we're not doing experiments on embryos, we're not doing experiments on babies, these are young adult rats. So how is this shRNAi targeting the specific gene. Once the plasmid enters the cell, it's taken up by this complex called RISC, which is RNA-induced silencing complex. And what this does is it monitors the RNA as it's being translated into protein, and it's watching for the one that we've got this bullseye on. So as it's going along, when it sees the gene that we've targeted, it cleaves it off, so that the RNA for the gene that we're trying to suppress now just kind of floats around in the cell and then eventually gets degraded. So the RNA is still there, we're just stopping it from being translated. So we've got a model for the knockdown, but can we use rats as a model to understand speech processing? Rats don't read, they don't speak English, they don't understand when you talk to them, even though they're cute and they're cuddly. Can they understand speech processing? I actually do have to go back to my computer for this point. How many people think rats can understand speech? Crystal knows the answer. I wouldn't have much of a talk if they could. So, this is a rat that's been trained to discriminate the sound dad. Pump up my volume here. Oh, where's my volume? Oh no.
Dad.
There we go. So she hears the target sound, she presses the lever, and she gets a little sugar pellet reward.
Sad.
She presses the lever to the wrong sound and gets a little timeout to say, no, that wasn't it. It is a long time. We really want them to get, this is not the sound that you press to.
Sad.
So now she knows, oh, that wasn't the sound, I'm not going to press this time. You're not going to trick me twice. Now the rat audiogram is higher than ours. So we shift the sounds 2 octaves up so that they can actually hear them. And so they're getting, again, is the correct hit to the target.
There's some science behind
All right.
cartoons.
That's right.
They have hyper choices.
So not only can rats learn that simple task, they can do a variety of other speech-sound discrimination tasks. They can identify English consonants, as you just saw. They can identify vowel sounds, so dad, dee, do. They can identify by the middle vowel. They can do this task in quiet, as you saw in the video, and in various levels of background noise. They can do the task after spectral or temporal degradation, which is a cochlear implant model. They can do the task in rapid sequences, so no space in between the sounds and presented very quickly. And they can also generalize across speaker and gender, so they can tell my voice apart from Tiffany's, and they can tell male voices from female voices. Here's an example of one of those tasks. This is the cochlear implant simulations. Each of these is a different level of degradation, which is important for the point I'm trying to make. What you can see is that as you degrade, as you go from right to left, the performance does slope down in both degradation methods. And we see the exact same performance function in humans. So they have basically the same thresholds for degradation, for noise, for speed, as humans do. And we can predict this behavioral performance by looking at the neural response patterns in auditory cortex. So how do we record neural responses in rats? We use microelectrode recordings, and we record activity from a small group of neurons. And by activity, I mean action potentials and local field potentials. How many people know what an action potential is? Most people? Okay, so I can breathe through the action potential demo. But for anyone who needs a little bit of a refresher, think about an action potential as a signal from one neuron to another. So if you think about 2 people talking on a tin can telephone, if we were to drop an antenna over top of them, looking for that signal as it crosses the wire, she says something in the tin can, it travels down the wire, gets picked up by our antenna, and then reaches the other person on the other side. For neurons, here's your neuron cell body. This is the first person in line. The axon of the cell is the telephone wire. And then you have typically hundreds to thousands of other neurons. They're down here waiting to receive that signal instead of just one lonely guy on the other end. So again, we insert our electrode into the brain, and as this neuron sends a signal down its axon, we pick up that signal via the electrode, and that's an action potential. When we talk about local field potentials, really instead of one neuron or a couple neurons firing, we're averaging across a larger group of neurons. So think about averaging tin can telephones across an entire town or entire neighborhood, rather than just 2 people. And this is what the action potential patterns look like when we put them all together. So there's a lot going on here. I'm going to walk you through what you're looking at. These are what we call neurograms. It's the average response of all of our recording sites to a certain sound. So let's look at the response to T here, and I'll just orient you to this figure. We've got time on the x-axis. Each row is a different site, and it's the average activity from that site. There are 445 recording sites in each of these panels. They're ordered on the y-axis by their characteristic frequency. In auditory cortex, each neuron likes to fire to a certain frequency the best. And so we figure out what frequency that is, and then we order them by their favorite frequency, so to speak. And when you do that, you get these really nice, unique patterns. And each consonant in the English language evokes a unique pattern of activity. So let's look at the sound B, for example. When you hear the sound B, these neurons that like low frequencies fire first, and then the higher frequency neurons fire with a little bit of a delay. To the sound D, you get the opposite pattern. These high frequency neurons fire first, followed by the lower frequency neurons a bit later. And we can use these patterns to figure out how easy a pair of sounds would be to discriminate, and we can also tell what sound was played by looking at the neural response patterns. And we do that with the nearest neighbor classifier. And so we train the classifier to look at two average responses to two sounds. So for example, here's the response to D, and here's the response to B. Once the classifier is trained to recognize these templates, we present it with single responses to one of the two sounds. The classifier calculates which template is most similar, and then it tells us, I think this sound caused that pattern of activity. So for example, in this case, here's our single trial pattern. Which one of the two do you think caused that? You are right. So in that case, it would be dad. And so if we look at classifier results for a variety of different pairs, we can actually predict behavioral performance. So the pairs of sounds that are easiest for the classifier to tell apart, such as D and S or D and B, are also the ones that are easiest for the animals to tell apart. And the pairs that are difficult for the classifier are also the pairs that are difficult for the animals. So we can use rats as a model for communication disorders because they can discriminate speech sounds with levels of accuracy that are very high, and they have thresholds that mimic what we see in humans. And we can also manipulate genetic expression. So the first question, how does a single gene, in this case KIA 0319, change the way the brain functions? So to find this out, we have to acquire neural recordings. And so we anesthetize the animals. We perform a tracheotomy to help them breathe throughout the experiment, and then we expose primary auditory cortex on the right hemisphere. We can lower 4 electrodes simultaneously, so we'll lower those into the brain, record neural responses to a set of sounds, move the electrodes to a new location, record some responses to sounds, move the electrodes again, and repeat ad nauseum. We can get up to 120 recording sites per animal, depending on how many sounds you want to play and who wants to stay up all night to do the night shift. These experiments can run up to 48 hours, and I've done them all myself before. They're not fun. So I was mentioning before characteristic frequencies. Let me show you how we get to those. So this is an example of a tuning curve. I apologize for the black. I'll orient you to this though. On the y-axis, you have intensity of the sound, and on the x-axis you have the frequency of the sound. So we play a variety of frequencies and a variety of intensities. Each of these little red dashes is an action potential. And you can see that at the loudest intensity that we played, this site responds to pretty much everything. You play something loud enough, everyone's going to respond. But as the sounds get quieter, you notice that this site is responding to fewer and fewer frequencies. And finally, when you get all the way down to the bottom, it's only responding to this cluster of frequencies. And so we define the characteristic frequency as the bottom point of this triangle. So this site's characteristic frequency would be somewhere around 6 kilohertz. When you're done with the map, you get something that looks like this. For the purpose of today, we're only going to talk about primary auditory cortex. And the sites in primary auditory cortex are arranged tonotopically, so that the sites that like these low frequencies are here on the posterior end, these middle frequency sites are in the middle, and the high frequency sites are on the anterior end. So you can tell where you are in the brain by how these sites line up and how the characteristic frequencies align. So for the first experiment, we took rats that were not trained at all. They were just born, raised in their home environments. We anesthetized them, and we acquired multi-unit recordings. 5 of them had the knockdown. You'll see it referred to as KIA minus in future slides, and 6 controls. We played a variety of auditory stimuli for them, the tuning curve, which I just showed you, broadband click trains, which are just broadband noise bursts that we present repetitively at different speeds. and isolated speech sounds that either varied by the initial consonant or the middle vowel. So before we talk about data, I just want to show you a couple more types of figures that you're about to see, so you don't get confused when they come up. This is the same EEG figure that I showed you before, but the local field potential figures look very, very similar. So when I talk about latency, I mean the time from the onset of the sound to this peak response here. When I say amplitude, I mean the space between the baseline firing and this peak response here. The other type of figure you're going to see, which you probably haven't seen before, is something we call a dot raster. And what that is the same site firing to the same sound 20 times. So you'll have time on the x-axis, each dot is an action potential, so this is one repeat of a sound, and here's all 20 repeats. So each row is a different repeat. All right, so now for the fun part, some data. So the first thing we wanted to look at when we started analyzing the neural responses from these guys is some kind of correlate to the human literature. Because again, we're trying to create a dyslexia model. We need to know if we're anywhere in the ballpark. And you rarely get a parent who's willing to sign a consent form for us to put electrodes in their child's brain. So you don't see action potential studies in humans. You see evoked potential studies in humans. So first we looked at the local field potentials. These are local field potentials from an awake animal and an anesthetized animal. What you see here is the same pattern we see in humans. The thin line is the knockdown. You see that the latency is delayed and the amplitude is lower, just like I showed you before in humans. And when you read about these kinds of results in human studies, they often refer to it as reduced activation or decreased activity in the brain. But without access to the action potentials, you can't ever prove that. But we have access to the action potentials, so we can prove it or disprove it. We expected to see fewer action potentials. So we counted the number of action potentials in the first 40 milliseconds of the response and the first 400 milliseconds of the response. And there's no difference. The knockdown animal's brain fires just as many action potentials as the control animal. So what's causing this lower amplitude? If it's not the number of spikes, then it must be the timing of the spikes. Because when you average, something must be causing it to go down. So we looked at the timing of the responses. Now in the control brain, if you play the same sound 20 times, the brain's going to respond basically the same way every time. You need to have a consistent neural response in order to be able to say, I know what the sound was. You've got maybe 4 milliseconds of error on either side. In the knockdown animals, we were closer to 30 milliseconds of error on either side. So the variability in when that site was going to fire is significantly higher in the knockdowns than it is in the controls. You can see this when we look at the neurograms as well. So on the top, these are the controls. These are basically the same patterns that I showed you earlier. Again, you can see the same patterns in DAD and in BAD, those opposite activation patterns. But now look at the neurograms in the knockdown animals. There's the exact same number of action potentials in those patterns as are in the controls, but the timing of them is very off. So even though it looks like there is reduced activation, there's really not. So let's look at some single site examples just to kind of drive this point home a little more. This is a control site. It's tuned to a high frequency. This is, again, the dot raster, so 20 repeats. Each row is a different repeat. And in response to the sound D, this site fires very quickly and very consistently across repeats. To the sound B, it fires a little bit later, but again, pretty consistently. And when you run this through the classifier, it only gets it wrong once. This one time where it fired way too soon. This is a knockdown site that's tuned to the exact same frequency. So you would expect this pattern, but instead you have this increased latency and this large variability in when those action potentials are going to happen. And when you run these through the classifier across all the pairs of consonants that we tested, the knockdown neurons are significantly worse at encoding consonant sounds compared to the control. This effect is not specific to speech, so I mentioned we played noise bursts to them as well. These are dot rasters for one of the noise bursts in controls and in the knockdown. So again, you see the same pattern, delayed latency, increased variability. And we quantified that in the same way. So again, longer latency, increased variability. There we go. So, knockdown of this gene causes higher trial-by-trial variability to tones, broadband noise burst trains, and degrades the neural encoding of speech sounds. So, does this neural impairment have behavioral consequences? We've seen a lot of times in rodent studies a neural effect that seems massive, but has no behavioral consequence. The brain is very good at compensating when things go wrong. And so it was completely possible that there'd be no behavioral problems in these guys whatsoever. But of course we have to test that. And so we trained 26 rats on a variety of speech tasks. They did the full speech task, which you saw in the video earlier. They did a speech and noise task, which was the exact same task, just in various levels of background noise. They did speech sequences, so it was sounds presented very quickly at a variety of speeds. And they did a truncated speech task, where we only presented the initial consonant, and I'll talk a little bit more about that in just a minute. So when we trained them on the full speech task, the first day everybody's kind of at chance. It's a difficult task. They have to learn what to press to and what to ignore. But you can see that the control animals learn this task very quickly, whereas the knockdown animals really are pretty sluggish about learning it. They're statistically significantly worse compared to controls on all 5 days. And it's not that they're hitting less to the targets, but they're hitting to the false alarms or the distractor sounds more often. So one thing we had to test, of course, there's a high comorbidity in humans between dyslexia and ADHD. So it was possible these animals were just hyperactive and just jamming on the lever all the time. And in rat models of ADHD, they've shown that the response times are significantly lower. As you would expect, they hit the levers faster. There was no difference in response times in these guys. So we don't think that they're hyperactive, we just think they're confused about what sound they're supposed to be hitting to. So then, of course, the next question is this due to the gene or is this due to the surgery? It's a pretty invasive surgery for a little embryo. And so what are we actually doing? When we did the surgery, we co-transfected with fluorescent protein. And all that means is that we could go back in the brains after the experiment was over and visibly see which neurons were affected and which ones weren't. And so we went back and we counted the number of neurons that had suppressed protein, and we correlated that with behavior. There's a very strong correlation in the knockdown animals, so that the animals that had more transfection were worse at the task, and the animals that had less transfection were better at the task. There's no relationship in the controls, so we really do think this is an effect of the gene and not an effect of the surgery. So we did notice, and I'll go back to the other figures so we can look at it, over the course of these first 5 days, that even though the Kia rats were worse, they were getting better over the course of the 5 days. So we gave them an extra 5 days to practice. And you can see that at the end of the full 10 days, they do eventually catch up to controls. So maybe this is just a learning impairment. Maybe they're okay at doing speech discrimination. They just need longer to learn the task. So we trained them on a difficult task that we know kids with dyslexia have problems with, and that's speech and background noise. And they had persistent problems on this task. This is the summary of the last day performance broken out by background noise level. And at the 3 levels that are doable, they're significantly worse compared to controls at all 3 levels. So then we thought, well, maybe they just have difficulty with challenging tasks. So we wanted to give them another task that was in quiet, that was equally difficult, to see, is it the noise causing them problems, or is it task difficulty that's causing them problems? I think I have time to show you the video on that one too. All right, so this is a slightly different setup. We have an infrared nose poke at the top this time instead of a lever.
Gad, bad, bad, tad, sad, bad, dad.
So when she hears the target sound, she pulls her nose out of the poke instead of pressing a lever.
Sad, sad, gad, tad, bad, tad, sad, gad, dad. Tad, bad, sad, gad, bad, tad, tad. Bad, sad, bad, tad, gad, bad, sad.
So she says, I didn't hear it. I'm still waiting to hear it.
Maybe she just really wants her dad.
Rat psychology at its finest.
Rat there.
Dad. Dad just means food. Dad means food. That's all dad means. All right. So we trained them on that task, and there were no deficits whatsoever. So it's a challenging task, and we played it actually at 4 even faster speeds than the fastest one I showed you, and they have no problems. So up until this point, you know, they've done pretty well with most of the tasks that we've given them. And we thought, well, maybe at this point they're compensating somehow. because these sounds are not synthetically created, so there's differences in duration, there's differences in pitch. This is a human speaking into a microphone. We're not perfect. And so maybe the rats are latching on to something that they can use as a cue. Rats are pretty smart. They can pick out little tiny things. And we know that kids with dyslexia are good at this as well. They can pick out prosody cues, they can pick out intonation cues to kind of help them get through their days. And so we wanted to see, are they really learning the task or are they compensating? So we chopped off all the extra cues and we only gave them the consonant sound. So it's a 40 millisecond stimulus and it's just the consonant. So the stimuli are now all the same length and the only thing they have is the consonant information. And they're horribly bad at it. So we think when they were in quiet, they were able to latch on to pitch differences, duration differences to do the task. But when you strip all that away, they never actually learn the consonant discrimination task. Now, there is significant improvement over the course of these 10 days. And we know that the current treatment for dyslexia is this intensive therapy on phonemes. The focus is phonemes. And so we thought, well, since they were getting better at it, could this kind of focused training also help the animals? So we took a brand new group of rats that had never been trained before and we started them on the truncated speech task. We gave them as much time as they needed to learn it. For controls, it takes about 28 days for them to hit ceiling, and that's exactly the same amount of time it took the knockdown to learn it. So again, this is an insanely hard task. I can barely do it, but the animals do, over 28 days, get better. And you can see over the last week or so, there's no difference across groups. So they can learn this consonant task, given enough practice, but does it generalize to the other tasks? We tested them on the full speech task. We didn't expect to see any differences, and we don't see any differences. But then we tested them on the speech and noise task, and now there are no differences across groups. And again, there's the breakdown on the last day. And just for fun, we tested them on the sequences task again, and again, there's no deficits there. So, rats with knockdown of this gene are impaired at phoneme discrimination tasks. Clear speech training does not help in adverse listening conditions, but this focused, truncated speech training does help. And the severity of the knockdown correlates to the severity of the impairment. But how is this training changing the brain, or is it? We know in humans, in the brainstem of children who have issues with language and reading, that the response to the second of two tones is weaker in the auditory brainstem compared to typically developing children. But after 8 weeks of speech training, you see an increased response in the brainstem to the second of two tones. We also see this effect in the reliability of brainstem responses. So at pre-test, the brainstem response from trial to trial is highly variable, like what we saw in cortex. But after language training, the reliability increases. So do we see this in the animals after training? So after 4 months of all this speech training, we anesthetize them, we map them. This is the same data I showed you earlier. The variability in the onset latency in the control animals and then in the knockdowns. Following 4 months of training, there's no longer any difference in the amount of variability in the onset latency in these animals. So we've significantly reduced variability in both the knockdown and in the control. So what does this do to consonant encoding? This is the classifier percent correct of the consonants in the untrained animals. So again, we saw that the key and knockdown brains were significantly worse at encoding consonants. Following training, there's no longer any differences. So we've increased the precision in auditory cortex by just purely behavioral training. So now to wrap it all back up and tie it in a nice neat bow, these are the LFP responses that I showed you earlier. So again, our analogy to human ERPs. Again, you see the delayed latency and the reduced amplitude in the responses before training. And after training, again, there's no longer any differences. So this suggests that we should be able to use current imaging techniques in humans to see these kinds of training effects. So 4 months of extensive behavioral training does significantly improve trial-by-trial variability in onset latency, and training also improves neural encoding of speech sounds, specifically consonants. So how does this relate to humans? How do we take this information and then apply it? We're proposing with this work that trial-by-trial neural variability is an underlying mechanism for dyslexia, and that children with dyslexia we know have inconsistent brainstem responses, but we don't know if it also happens in cortex. Is this something that is systemic throughout the auditory system? And so my future work here, hopefully at the IHP, will investigate this hypothesis in kids with dyslexia, SLI, and kids that are comorbid for these disorders, and also look for any link between variability in the brain and a specific genetic variant. Hopefully KIA 0319 will make an appearance in that stuff as well. So this work was done at University of Texas at Dallas, and I couldn't have done it without my army of undergrads, my colleagues, my mentors, and of course the rats. So thank you for your attention. I'd be happy to take questions if you have any.